Short Bio Labs, I founded it in 2005, and today we are, I was myself, and then 6 months later hired an employee. Today we have 14 employees. We do a whole bunch of different things. We have laboratory testing, so we're an accredited ISO 17025 laboratory. We also have a field application side of it where we go out and do a lot of testing. As Craig was mentioning, we go out, investigations, a lot of commercial buildings, residential, industrial manufacturing, all kinds of stuff. Any place there's a microbial problem indoors, which we call the built environment, we deal with it. So we ended up getting into cannabis. It was cannabis first, and that was, I guess, about 4 or 5 years ago, where we got some calls from out west in illegal states, and one of the companies was, you guys may have heard of them, LiveWell. They're a really big company out in Colorado, and they had set up some new facilities, and they were all indoor grows out there. So most of the field work we've done has been with indoor grows. They tend to be more problematic for certain microbial issues. But anyways, so we started working with them, and we came up with a method for monitoring, using some more technology and things like that. Obviously you can't send any material back there, but they have a really good staff. They have a lot of PhDs on staff. I mean, it's a really high quality operation. And so we taught them how to be able to do an extract for test for mold off of plant material and send it back to us, make sure it was clean, there wasn't any plant material in there. And we taught them a couple other different things, and DNA extractions, things like that they could send. So we do a lot of DNA testing too. From the DNA standpoint, we test for microbes. So we can tell them without having to have actual, with just DNA extract from a material, we can tell them what microbes are present. Anyway, so I'm going to talk about different things we do and some of the stuff that we've gotten into on the hemp side. So, Steve mentioned before, the thing with hemp and cannabis, just get right on this thing. So, you look at the plants, you can't tell the difference. This is LiveWell's facility in Colorado. This is a Bluen's facility in Knoxville. So obviously this is cannabis, and this is hemp right here. You look at these plants, they look the same. Most people, I think, have confusion about that, but that's one of the things that you can look at this stuff. That's why I think a lot of the law enforcement's having a big issue with flowers right now. It's like, well, is it hemp or is it cannabis? I mean, they don't know. So some of these groups are trying to get things outlawed of being able to have flower, which I think is a big issue. They need to come up with some tests so they can actually measure these things in the field. So the services, from the service side, we have facility sanitation reviews. So this is a lot of stuff we do out west, or these indoor grow facilities. There's a lot of things with hygiene, just how things are functioning. You have a big hydroponic operation like LiveWell, where they have 66,000 plants in a building. There's a lot of water moving around, a lot of nutrients, a lot of other things going on, a lot of air movement. A lot of these things, that can, if it's not balanced correctly, it can have a lot of microbial issues. And since all the plants on the cannabis side, it's mandated in those states by the state that they have to be tested for mold, yeast, and bacteria and some different pathogens. Obviously, they have to make sure that they're not having contamination on their flowers. If it's contaminated, if it's above the threshold after the test, they have to destroy the whole batch, which is a lot of money for these guys. And also, from the whole standpoint of, it's not just the product. In product, you have workers who are working with all these things. So when you go into these locations, you have grow rooms, big grow rooms, but then you have the processing of the material, where you go in there and there's people in there who are putting taking the flowers and basically have machines that grind it up and put it into cigarettes. So they're doing all this stuff, and then you have a lot of dust generation and things like that, and all this stuff gets airborne. So you go in there, you're looking at 50,000 spores of mold per cubic meter of air. That's just massive. Nobody wants to be breathing that. So there's a lot of health and safety concerns also, because you don't want your workers getting become atopic or sensitized to this stuff. You can have a lot of issues. So the whole hygiene thing and health and safety There's a lot to it. Facility mold and bacteria monitoring. So we do monthly monitoring in several of these facilities out west, where we're actually capturing air samples out of the air, and we look at just several different things. We look at different types of molds that are important for contamination purposes, so they don't fail, and also a couple different pathogens. But the primary one is powdery mildew. It's a big problem on indoor grow facilities. So these facilities will monitor throughout their locations, and if they get any it's a monthly thing, so if they get any hot spots, they're able to go back in, okay, what's going on? Let's look at this and readjust our SOPs and things like that to make sure this isn't happening. Excuse me. So from the sanitation side, we want to maintain a facility that minimizes microbial contamination, and that requires, identifying the sources in the facility. We need to know what genus or species level at different differentiation to assess risk. Some things are greater issues for like health, aspergillus and penicillium. Species of mold are more of a health concern. Small spores get into the lungs, they produce toxins, all those things. And also, they're prolific. So from that standpoint, it's like I know something that can just produce a massive amount of spores really quick. That can obviously cause problems for my employees, but also cause problems for my end product. So we work with them to develop a facility hygiene plan, and that'll include, all these different things that we find during the investigation. And also, it includes things about their health and safety plans also. So we work on both sides of that. And we also do the chemical side for facilities that have that do extractions within, like Livable does a lot of extractions inside in their own facilities. So they have chemistry rooms, and they're using a lot of different solvents. So we're not testing those products for solvents, but we test the air. So there's standards out by OSHA, and where we do TO15 canisters and things like that in order to make sure that they're protecting their workers, they're not having leaks out of their hoods and stuff like that. Talk about the monitoring, and then we do annual audits on these things and do updates where needed. So we have all the data, we can look at things and help them get better, enhance their quality over the years. So let's talk about microbial testing in general. Now this is for, like all the states I said that have medical marijuana or legal cannabis for recreation. They all have to have these things tested, and there's on the bacteria side, we have E. coli and Salmonella bacteria. Well, this is I think this is out of Colorado right here. And then we have total yeast and mold, and the different concentrations there. And this is across a variety of things: flours, retail products, concentrates. You guys have probably seen a lot of these different things they have. I mean, it's just a massive variety of different types of products. So for hemp, what we're doing is we're set up and we're running these already. We've been running processing samples, I guess, for about 2 months now in the lab for the mold side of it. We have some standard panels that do your basic mold and the bacterial concentrations and yeast. We have some expanded panels. These things mimic what we see out west. So those offerings, we want to have like the basic thing you need to do, and then also like if you look at California, obviously the state of California always takes things to the nth level, and so they're kind of like they'll have like the greatest number of tests that's mandated to do on these things. So we cut across the gamut of those things that we can do in the laboratory. Different types of samples, matrices we can handle right now: flours, extracts, oils, lotions, and other bulk samples. We're not doing any gummies or anything like that yet. Those matrices are difficult for microbiology. We just haven't moved into that. We are going to move into that, but there's just not the need yet. So some of the basic things here, which is total bacteria, which would be aerobic bacterial count. We have the analysis, incubation times, and our levels here. So you see it's based on a weight basis. So for like aerobic bacteria, which would be total bacteria, takes about it takes 48 hours before we have a result. So things do have to grow when we culture them, and you have to be under 100,000 CFUs per gram. Coliforms, these are things that are found in the feces, things like that we don't really want any of that in our sample, but we see different levels of that. So it's a much lower threshold with 1,000 CFUs per gram. Total yeast and mold count at 10,000 CFUs. These are colony forming units per gram. And then when you get into the nasty things like E. coli and Salmonella, so E. coli is a coliform, but it's a pathogenic coliform, and you see that it has to be less than a CFU. So you can't have any, you have to be basically a non-detect. So you have, sample size, 3 grams sample, 5-day turnaround on like the yeast and mold, that's 5-day. Bacteria come out sooner, and then that's, this standard panel here is a $50 panel. We also do foreign matter, so all the samples come in, we look at foreign matter, and that's incorporated into this. And so we see different things like spider casing there, some different fibers. And that's looked at, and it has to be under 5% of the sample. It has to be foreign matter, which is usually not a problem, unless I don't think we've seen anything that had that much in it, over that percentage. I don't envision it happening if you're doing your processing and there's somebody doing machining or something else and there's material getting in there, some kind of other manufacturing going on in the same facility. Additional tests, we have the O157, this is the flushing E. coli, and we can do that per sample, and then we also do the powdery mildew, this encompasses all powdery mildews that can affect hemp, and that is 75% per sample, and this is actually a DNA test here on the powdery mildew, and so we have our turnaround times on those. And we're working some other pathogens in as we see them in the field and they become issues. So this is indoor grow here, and we work with Blue, and they send samples in for things along these. They're a big company, a German company that's set up in Knoxville. They do extractions. They have some state-of-the-art facilities to do this type of things. They're also doing their own grows. So they're doing indoor grows in Knoxville. So on the hemp side, so we're working with them on sanitation and storage. Storage is a big issue. You got to maintain things in storage. You got to dry stuff down, get it to a certain moisture level so you don't have a bunch of mold and bacterial growth when your product degrades. And they make all kind of products over at that company. These are some free hemp cigarette samples they gave us. They were processing, making all their own stuff there. Here's some of the different types of products I was talking about. This is out in Colorado, and you see I think it's a bunch of live well stuff here. All kinds of interesting things that we deal with on that side. They have to deal with testing. So, like I said, it's not mandated for hemp yet, for mold and bacteria on these products, but we recommend that you do test. Obviously, we're a lab, and so recommend you do test. But the thing is, I think consumers are gonna demand that type of testing, even if it doesn't get to the regulation point, because nobody wants to be smoking moldy flowers because they have all the mycotoxin issues and health issues. And you just look at something that you open up and it's moldy, it's just a bad product. It's going to smell bad. It's not going to have the quality of a really clean product. And you don't want to breathe mold in your home. You don't want to breathe it in when you're smoking this product or consuming it. So on the sanitation side, some of the things that we see when we go into these grow facilities indoors is simple things. You have water condensation on the drip feed systems, on the PVC pipe. Anytime you have water, free water like that, biofilms are going to form. You're going to have a bunch of mold and bacteria growing on that. That's going to get on your product. One of the big things we see is that all of the detritus, all the leaves are falling down on this mat. The mat's wet. Well, that's just massive amounts of mold grow in there. So a lot of stuff is like we get into a facility that hasn't had sanitation done before on it, it's, okay, you just need to go into your SOP so you can get a big fix right away. It's like, well, we can work this really easy because we have people walking through these things every single day. Well, they're going to pick up the leaves that fall on the ground. They're going to do that as part of their process when they're looking at stuff. It's not much extra work, but it's a huge improvement in indoor air quality and reduction in mold in these locations. These are biofilms that are forming on the pipe. I mean, it doesn't look like much, but when you get down to that, you know, an area like this can have 1 million spores in it very easily. So, you know, the things that you don't see they aren't really evident that can get you on this side when it comes to microbiology. You see biofilms are growing on the pipes, so they're all, you know, the moisture rolls down the side of the pipe and it forms here and you get drips and things like that. So you have a lot of biofilm formation. So simple things like wrapping these pipes, it was an easy fix. You stop the condensation. Some more pipes and then on the mats, you have a lot of biofilms and detritus. Just clean this up when we come through every day. It's not a big deal. It's like anything in any industry we work in, whether it's healthcare, it's cosmetic manufacturing. A lot of times it comes down to the cleaning side of it. You clean your mop after you're done with it, you throw it in a corner, let it sit there and ferment overnight, and use it the next day to just move all the microbes around again. You know, it's like just some of the basic things you need to think about when it comes to hygiene. Wood, kind of take out all the wood. You don't want wood on your near your mats because it's just going to suck moisture and grow a bunch of different types of molds. You know, again, the detritus, I mean, you should never see this on a mat. This should have been picked up days ago before it had a chance to decay and rot there. There's some more detritus and wet. Want to keep water off, so one of the things with a lot of these mats, when they were installed, they weren't balanced right, so the moisture, the water runs off. And they have drain systems and everything, but it just wasn't, they weren't set up correctly. So we had the engineers come back through and set everything up, so we're pulling all the water off. I mean, this is just a mess. This is an early veg room, and it's really hard to take photographs in these rooms because they have all these LED arrays at different different spectrum for these types of rooms, and the mother plant rooms are different. But anyways, these plants are all here, and they're pulling them, they're moving them to another room. And it just covered with detritus all over here. And, so that doesn't happen anymore in this facility. They cleaned all this up, and their concentration of issues and contamination and the amount of batches they lost because of mold and bacteria just dropped. I mean, significant drop. Like it was an 80% drop in their loss. A lot of the testing you do is for viable, so most of the time it's for viable. So we look at the different things, and Aspergillus, we have some penicillium here, Cladosporium. These three are the big genera that we're usually looking for. Here's Aspergillus penicillium on an air plate, a ton of colonies in an area that had just a lot of detritus on the mats and a lot of sporulation going on. Air moving systems, one of the big things for these big grow facilities that move a lot of air. This facility had to move, so they had on top of the building RTUs. I think they had 64 RTUs on top of this building, moving just a massive amount of air. So much air that they were having when they first put it up. There was doors that were closed because of the air movement, and they could not physically open them. So they actually cut. This thing's this two-level building. So between the two buildings and it's cement floors, they actually cut through the cement floors to get the airflow to circulate, so they can open their doors up because they were getting some major vacuums in locations. So ventilation is pretty important. On the ventilation side, you need to have air movement. This canopy gets thick, and this is where a lot of disease develops down in this bottom part of the canopy. So they move a lot of air. They have fan systems in. Obviously, you have a lot of lights. You have a lot of moisture, a lot of things going on here. You want to keep that air movement, helps to evaporate, get it out, and they can move it out with the air scrubbers. and the air RTU systems. So they need to get that lower canopy, have some air coming through it, or they're going to have a lot of powdery mildew and other mold issues. We have some products that we developed, some technology that's really has high capture efficiency that you sprayed onto these filters. So a lot of these guys were using a bunch of these systems where you have these HEPA filter units throughout the building that capture the mold and bacteria. HEPA filters are very expensive, so we're able to use this solution we call molecular entrapment, and you can buy a cheap filter and put that on there, and you don't get the dust capture, but you're getting all the microbial capture. There's a special protein in there that is really good at capturing mold and bacteria and different viruses. In the pathogen monitoring, there's a powdery mildew issue right here on this leaf. Lower canopy, probably not a lot of air moving through there, and again, we have a lot of moisture issues in here. Here's some more powdery mildew. Powdery mildew won't outright kill your plant, and powdery mildew is what they call an obligate biotroph, which means it has to have a host. You can't grow it in media, so when you have your stuff go to the testing lab, they will never be able to tell you if there is powdery mildew if they're doing a viable analysis on it, because it won't grow. It has to have a host to grow. But from the standpoint of yield, when you have all these big fan leaves covered with powdery mildew, well, all my photosynthetic areas just get knocked out, so they start to grow a lot slower. They don't have as much energy, and then they're more susceptible to nutrient deficiencies and a whole bunch of other things. It just, they can go downhill pretty quick. On the back side, we saw the top of the leaf where it's powdery, but if you look at under the side, this is powdery mildew. On the underside, they produce these little things called Cleistothecia, and this is where you have, on the top side, it's asexual reproduction, producing a bunch of spores that spread. On the underside of the leaf, they produce these Cleistothecia, and this is all sexual reproduction, so you get new genetic variants here. So it's one of those things that gets in there, it mutates, obviously everything mutates over time, and you get some really nasty strains of powdery mildew that just cause a mess in your crop. That's they're really cool looking. On the standpoint of what we do, so we actually did a grow this year at our laboratory, outside grow, so we got our permits from the state. And one of the things is I've been working for the last couple of years with a group up in Massachusetts. We developed it's a parasite of powdery mildew. So we wanted to be able to, this is the first year of our project. I've been sending off material to them to test it on and do trials, but since we're actually can grow, and it's the same darn plant, hemp versus cannabis, just the THC concentration. So we were able to set up trials, and this first year was just to see if we could actually grow the plants. And so we just a small, I think we had 40-something plants, and so we got went through all the paperwork, learned how to do all this stuff, and dealt with the state. It wasn't too difficult. I mean, for us in Tennessee, I don't know how it is here, but you have to have a GPS location. You have to send them coordinates with a photograph. So this is our laboratory here, and this is our grow area over here. We used air pots. It's made by, I think these are made by a Swedish company. They're really cool pots in that they have these holes in them. And what happens is the roots migrate to the holes. They're pruned because a root needs to have, pretty much a low oxygen. When it gets high oxygen, it'll actually kill the root. And when it kills the root, root tip, it prunes it and it splits. So you get massive, awesome root. The roots are just amazing on these things. So these are air pots, and if anybody wants to know about that company, we can get you information on it. But we use these air pots. We use some other ones also, just the bags to grow them in. Here were our germlings back in May. Got these started. We had 2 strains. These strains, they have all kinds of names. One is Special Sauce, and the other one's Hawaiian Haze. Those are our 2 strains. So here's some of our plants in the mid-season. I was surprised we had good growth. It worked out pretty good. I didn't know what to expect, but we watered them daily. We fed them liquid nutrients. We used just a simple, basic soil. It actually was shipped. It's a California soil, but there's all kinds of soils you can buy. This is just a really cheap, basic soil from California, and it did a great job. It was really well-drained soil. If anybody wants that information, let them know. So throughout the season, this is the first year. We didn't do any trials with the powdery mildew parasite this year. We just wanted to see if we can grow these things. And so we documented a bunch of other stuff, like we have stink bugs here on our plants, which is, you don't want stink bugs on your plants. But we also have found beneficial bugs, like ladybugs and assassin bugs. And this guy was one of our favorites. He hung out and he really tore up all the nasty insects that came in. That's a green lynx spider. So we talk about some of just our experience, my experience with testing and what we had to do for the process. So we talked a little, I think Steve talked earlier about testing. We wanted to see where we were with our samples, with our plants, as we were probably like in the third week of flowering. We wanted to see, okay, where are we? What's our concentrations? And one of the things we do is we have dissecting scopes in the lab, so we take these off and look at the trichomes, and you can see when the trichomes aren't milky, they start to clear up. And you can probably do it with a little eye loop too, but when the trichomes get clear, you're getting closer and closer to them being ready for harvest on that standpoint. That's just some visual observation you do. It doesn't tell you THC or CBD concentration, but it's telling you that it's something that's been done for cannabis for years and years. Are my trichomes, is that liquid in there milky or is it clear? When it gets clear, you're getting there. You're about ready. And I wanted to know basically what's a concentration. So you hear people taking pieces from here and here and have leaf material. Well, we want to know what the bud material was. That's really important for us. And so we actually trimmed and everything. This is a trimmed bud that we picked to send in for analysis. So we wanted like the optimal specimen. And we did this from several different plants and several different locations. This one here came back. We had a Delta 9 of THCA of 0.1, no Delta 9, I guess, or CBD. CBD is 2.53 on that. That was from the special sauce strain. Our Hawaiian Haze is right on the edge. Let's see, on the 0.29 on the Delta 9, but higher CBD concentration. So this is probably about 4 weeks ago, 3 or 4 weeks ago. So right now is actually when we're gonna leave here soon and head back, and we're gonna harvest all these plants tonight and hang them and everything up tonight and tomorrow. They're due. So what we did was 2 weeks ago we had this state inspector come out, take samples so we can get our results. Well, Tennessee, from what I understand now, they have massive backup on these things. And so they said we would know within 2 weeks. Well, we didn't. It's been 2 weeks and we didn't know. So we're calling the state. I mean, we're at the point where we're going to start degrading now. So we call the state 2 days ago. They give us a verbal, say, okay, you can cut them. They don't know our concentration yet. You can cut them, but you can't sell them. At least we can cut them. And so I had one of my guys today call and specifically asked the inspector to send an email to say that we can cut them.